Hi, my name is Himit Denoa, and today I'm going to be teaching 3 levels of genetic mutations and their effect on enzymes. Today I'll be talking to first a young child, also known as Catty. I'll be talking to a teenager, Pineapple, and lastly I'll be talking to a current Harvard student, Fan. Thank you, and I hope you enjoy. Hi. Today, I will be talking to my young friend, my 12-year-old Catty. First, I'll be talking to him about what an enzyme is and how it relates to genetic mutations. Hi, Catty. An enzyme is a special thing in your stomach. Enzymes are responsible for a lot of the work that is going on in the cells. They act as catalysts. And I know you're thinking, catalyst, that's a big word. But it's pretty simple. A catalyst they act as catalysts in order to speed up important chemical reactions in our stomach. For example, when you drink milk, your stomach needs to get the nutrition out of this. To do this, our body uses a special enzyme called lactase to digest milk and break down lactose. However, you must remember there are millions of enzymes in your stomach, and each enzyme has its own job. For example, your dad is a doctor and he helps people, and your mom is a banker and makes money. Each person has their own job and role, and enzymes are the same. There are things that can affect enzyme activity. For example, temperature can affect the job of an enzyme. It's like getting sick. When mommy is sick, she may not be able to work at all or may work slowly. Same thing happens to enzymes. The colder the temperature is, the slower the enzyme does its job. However, one thing that can cause enzymes to also stop working are genetic mutations. Mutations can occur anytime, and these cause the enzymes to transform. Imagine your dad's a doctor, and his arms randomly become squid arms. It'll look cool since he has squid arms, but he can no longer do his jobs. Genetic mutations do the same and stop the work from happening, and this is bad if it can stop enzyme activity, because enzyme activity is so important in your body. And if one job stops happening, many other jobs may stop as well inside your body. Now, I'll be talking to my teenage friend, Pineapple. Say hi, Pineapple. Hi. Hi, so today I'll be talking to you about enzymes and genetic mutations. Enzymes are biological molecules that speed up the rate of chemical reactions within the body. These are vital in our body for various functions. It can help with digestion, breaking down food, and breaking down different substrates of that, meaning it can break down protein, break down carbohydrates. There are countless things that enzymes are needed for. Some enzymes help break large molecules into smaller molecules that are more easily absorbed by the body. Enzymes are like keys. They have a special area, an enzyme. Like look at this, for example. There's a special area, only this area where the candles can get lit from. An enzyme is the same. It is like a key. The active site is the hole in which the rope of the candle goes into. If the candle will never get lit unless the rope is put in the right place and then lit. The same way enzymes work. Enzymes are like a lock and a key. The right shaped key must go into the lock of an enzyme, which is also known as the active site. There are only 2 ways in which you can regulate the enzymatic function, and the regulation is important, or else the same key can just keep on going in and out and doing too much. And in order to make sure it doesn't do too much, there are 2 main ways that it can be prevented. First is competitive interactions, in which another molecule, imagine another rope which can't get lit on fire, binds to it. Another thing binds to the enzyme, and this is a competitive interaction. Also, it doesn't always have to be as direct as that. There can also be non-competitive interaction, where a regulator binds to another area of the enzyme, causing the active shape to change. So imagine the rope that you were constantly putting into your candle just to get it lit on fire. A knife comes and breaks that hole and changes the shape, and the rope no longer fits. That's the same thing. There are also many factors that impact the speed at which enzymes can work. Firstly, temperature has a large effect. The warmer the temperature, the faster the rate of reaction will be, and the opposite for when it's cold. pH can also impact an enzyme. An extreme pH, high or low, will typically slow the reaction or stop it. And concentration as well. The higher the concentration, the higher the reaction rate. And now I'll be relating this to genetic mutations. A change in the DNA that is inherited through cell division is a mutation. Mutations can impact protein development. Nonsense mutations occur when a codon is converted to a stop codon. Think of molecular code like a train. Imagine a train is a long train with multiple coaches. The way nonsense mutations work is, imagine there's two trains, and then you cut off the thing connecting the second and third coach. The third coach and all the trains on will no longer work and will be stuck behind. This is the same way a nonsense mutation works. A mutation changes a normal codon to a stop codon, which causes everything else to also stop working. And a mutation like this impacts proteins, and enzymes are proteins. A mutation can do lots of things. It can alter the active site, the area which I said the candle's rope goes directly in, and the specific substrate will no longer fit. And this can cause the enzyme to no longer be able to work and do its job. And this is very dangerous, as enzymes are integral to our body's function. And genetic mutations can cause these functions to no longer work. Hi, now I'm talking to my college friend, Fan, who currently attends Harvard Biology. How's it going, Fan? Today I'll be talking to you about enzymes, and more specifically a genetic mutation that can cause an enzyme to no longer work. You're a very smart guy, so you already know the basics of enzymes and genetic mutations. You know that enzymes are biological molecules that speed up the rate of chemical reactions within the body. And you know these are vital for our body for various functions. Some enzymes, help break down large molecules, while some enzymes do much smaller jobs. Enzymes have a special area called the active site where substrates, the reactants, enter. When the substrates enter the active site, the enzyme does its job. It can break and form particular bonds. The shape of the active site of an enzyme is shape specific for its substrate. Genetic mutations can significantly alter the function of an enzyme. When a protein enzyme is being built, they have a specific code. However, different mutations can cause the code to be altered. And I'll be talking to you about a real-life example. This is the maple syrup urine disease. I know, it's a funny name, but it's a real thing. Valine, leucine, and isoleucine are amino acid chains that are present in many kinds of food and are broken down in our body by an enzyme called branched chain alpha keto acid hydrogenase complex. This complex is formed by multiple copies of three types of enzymes referred to as E1, E2, and E3. The human genes encoding the proteins that constitute the E1 component of the complex have been found out to be the location of many identified mutations which have caused MSUD. MSUD is caused by mutations in one of three different genes: BKCHDA, BKCDHB, and DBT. Mutations in these genes result in absent and decreased activity of the human branched dehydrogenase complex enzymes. So what this causes is the amino acids which the enzymes help break down, there are not as many of those amino acids being broken down, and therefore there is an accumulation. And an accumulation of this has also leads to accumulation of their toxic byproducts, keto acids, which result in serious health problems associated with MSUD. And the toxicity of these amino acids are very bad for the body. A simple mutation in the E1 enzyme can cause such negative side effects to the body. It can be as small as a simple headache. However, it is very common for people that have experienced this genetic mutation to have seizures and ultimately faint, which is why it is important to have an understanding of enzymes work and what mutations can do, and have a scientific understanding of how we can potentially mitigate these errors.